Welcome to the deep dive. Today, let's think about the human genome, that incredible instruction manual for life.
Right.
Now, most of us get what a huge achievement it was just to read it, mapping everything out.
Absolutely monumental.
So what if I told you there's now this really groundbreaking project aiming to actually write it from scratch?
That's exactly right. We're talking about the Synthetic Human Genome Project, or SynHG. It officially kicked off in 2025.
SynHG. Okay.
And its mission, essentially, is to synthesize human DNA molecule by molecule right there in the lab. It's genuinely a world-first attempt to artificially create, well, the basic building blocks of human life.
Wow. Okay, so our mission today then is to unpack the science behind this, the incredible innovation, explore what it could mean for medicine and, everything else.
And also the ethics.
And definitely examine the significant ethical questions, yeah. And also this unique approach they seem to be taking to oversight. We want you to leave this deep dive really understanding why this is seen as such a paradigm shift in biology.
Definitely. And to do that, we've looked at a whole range of sources, foundational documents, reports from groups like the Wellcome Trust, GenomeWeb, plus research from leading universities.
So solid grounding.
It's a deep look, yeah. This could be one of the most important scientific efforts of our time.
So we've got this idea of writing life. Let's look closer at how this builds on what came before. The Human Genome Project, finished back in 2003, that was all about reading the map.
Exactly. Reading the instruction manual, like you said.
But SynHG is different. It's not just reading, it's synthesizing, creating these sequences entirely in the lab, molecule by molecule.
And the sheer scale of it is, well, it's pretty astonishing. This isn't just some small research grant.
No.
It's explicitly called a world-first attempt for human DNA synthesis, and it's backed by a pretty significant £10 million from the Wellcome Trust.
£10 million, that's serious commitment.
It signals huge belief in the potential, yeah.
So, okay, it's ambitious, it's well-funded, but why now? What's driving this acceleration? It feels like things are converging.
You've hit it exactly. This isn't happening in isolation. It's really emerging right at this point where we've seen huge leaps in AI, in robotics for assembly, and in synthetic biology itself. They've all kind of come together.
Okay, tell me more about the AI part. You mentioned EVO2.
Right, EVO2. It's an AI model trained on, get this, over 9.3 trillion nucleotides.
9.3
Yes.
trillion. That's just, I can barely comprehend that number. What does that let it do?
Well, it means the AI isn't just finding patterns anymore. It's starting to understand the deep grammar, the language of life, you could say. These machines can now effectively read, write, and think in nucleotides.
Think in nucleotides.
Yeah, predicting how genetic changes will actually play out biologically with accuracy we just haven't had before. This ability, it's expected to completely transform how we understand genome biology and, of course, change the game for biotech and medicine.
So it moves beyond just seeing correlations in genes to understanding actual cause and effect.
Precisely. You can intentionally design and build specific genomic pathways to see exactly what they do, how they contribute to disease, for instance, something traditional genetics, which mostly observes, couldn't really achieve in the same way.
Mind-boggling. Okay, so looking back a bit, how did synthetic biology even get to this point, where synthesizing a human genome seems, feasible?
It's been an incredibly rapid evolution, really building on decades of molecular biology and genetic engineering work. We saw those early successes, like synthesizing genomes for simple bacteria, Mycoplasma mycoides, E. coli.
Right, the bacterial genomes.
But it's not just about copying what's already there. Researchers have even created completely artificial nucleotides, things like 3-ferranyl nucleic acid, or TNA.
TNA. And why is that important? What's special about an artificial nucleotide like that?
Well, work from the University of Cologne showed TNA is way more stable than natural DNA or RNA. And crucially, our cells' degradation enzymes don't recognize it.
Ah, so the body doesn't break it down easily.
Exactly, which means you could design genetic material for, say, therapies that last much longer, or even for biological data storage. It bypasses some of our natural defenses. That kind of fundamental innovation helps pave the way.
Incredible. So the SynHG project itself, how is it structured? Is it one central lab or?
No, it's highly collaborative, a 5-year, multi-center effort. You've got researchers from Cambridge, Kent, Manchester, Oxford, Imperial College, London, all involved.
A real powerhouse group.
Yeah, and it's led by Professor Jason Chin from the MRC Laboratory of Molecular Biology.
And I heard Professor Patrick Yeezy Kai at Manchester is key on the tech side, using AI and robotics.
That's right. Professor Kai really emphasizes using integrated generative AI, these advanced robotic assembly platforms. It's all about revolutionizing the process, making it scalable.
Scalable synthesis, so making bigger changes than we could before.
Yes, much larger scale changes than traditional gene editing allows, which again is key for figuring out those causal links between DNA sequences and actual human traits or diseases. And of course, they're building in rigorous quality control all the way through.
Makes sense.
Yeah.
So they have this big vision, but how are they tackling it? Is there a phased approach, a timeline?
Absolutely. It has to be phased. Phase 1 just started June 2025. This is the foundational 5 years.
Okay.
Focuses on developing the tools, the methods, learning how to synthesize progressively larger chunks of human DNA. They'll make small controlled changes to chromosome sequences, ones that shouldn't have big effects on protein production, just to hone the techniques.
So building the toolkit first. Makes sense. What's phase 2?
Phase 2, that's years 5 to 10. The goal there is ambitious. Build a complete synthetic human chromosome.
A whole chromosome.
Yeah, it's about 2% of the total human DNA, but it would be a massive proof of concept. Show it can be done on a significant scale.
And a long-term dream.
Yeah.
Decades out.
Complete genome synthesis. This current project is really about laying that essential groundwork, building the foundation.
Okay, with that kind of power, the potential applications, let's talk about the promise, especially in medicine. What could this actually do for treating diseases?
Oh, the potential here it's hard to overstate. In medicine, think about generating cells that are resistant to certain diseases.
Disease-resistant cells.
Yeah, using them to repair or even replace damaged organs, liver, heart, immune system components. You could engineer these synthetic cells to fight off specific illnesses or even have enhanced regenerative abilities beyond what natural cells do.
That sounds almost like science fiction. What about something like aging?
Well, Dr. Julian Sale talks about future therapies that could improve health span. Not just living longer, but living healthier for longer, with fewer age-related diseases.
Whole span, right.
And then there's personalized medicine, using synthetic nucleic acids for super-targeted drug delivery, hitting only the specific organs needed, or using them in diagnostics to spot viral proteins or disease biomarkers with incredible accuracy.
Beyond the human body, what about other areas, like agriculture or the environment?
The ripple effects could be huge there, too. Think about agricultural innovation engineering crops to withstand drought or extreme temperatures. That could be massive for food security.
Especially with climate change.
Exactly. And potentially contributing to defending biodiversity using synthetic genomics. Plus, industrial biotech, designing microbes to produce biofuels efficiently, or manufacture pharmaceuticals, or even clean up pollution.
And for the basic science, the fundamental research.
Yeah.
How does writing DNA change how scientists actually study life itself?
That's a really key point. When you build DNA from scratch, you can truly test how it works. Test theories in a way that's just, well, difficult or impossible if you're only tweaking existing DNA inside a cell.
You can isolate variables more easily.
Precisely. Create very specific genetic setups to see what happens. This also means much more accurate disease modeling. You could build the exact genetic architecture linked to a disease and study it directly, speeding up drug discovery.
Okay, the potential is obviously enormous. But this brings us to the other side of the coin. The peril. The ethics. This has got to be stirring up debate.
Oh, absolutely. Huge debate. One of the first things people jump to is the designer babies idea, right? Human enhancement.
Yeah.
The fear is this tech could lead down a path to creating humans with specific traits or making unpredictable, maybe harmful changes that get passed down generations. It raises really fundamental questions about human identity. What's acceptable to change?
And what about the potential for misuse? Dual-use research.
That's a major concern, too. Could this knowledge be used to create biological weapons or enhanced soldiers? Or even strange hybrid creatures with human DNA? Serious security worries there.
And the practicalities, like who owns this stuff?
Exactly. Who owns a synthetic organ? Or, hypothetically, a synthetic human. Who controls the data generated? These are massive legal and commercial questions.
Some experts sound really worried. Professor Bill Earnshaw's comment, the genie is out of the bottle. That's pretty stark.
It is. It reflects a view that trying to completely restrict this kind of powerful knowledge might be futile in the long run if some group is determined enough.
And the biology itself is just so complex.
Right. Biological systems are incredibly intricate. Making big synthetic changes could have knock-on effects we just don't foresee until it's too late.
Which brings up social justice too, doesn't it?
Definitely. If these therapies or enhancements are incredibly expensive, could they just widen the gap between the rich and poor? Create genetic haves and have-nots? It's a serious worry.
And we've seen ethical breaches before in genetics. The Hejiankui case with the gene-edited babies.
Exactly. That was a huge wake-up call about the risks of rogue actors and the absolute need for strong ethical oversight.
Which, interestingly, seems to be a really central part of this SynHG project, their approach to responsibility and oversight.
Yes, and they seem to be trying something genuinely different, trying to build a new model.
So, given all those really deep ethical concerns, what is so innovative about how the SynHG project is handling it? How is it different?
Well, I think the key difference is integration. They have this careful synthesis program. It's not an ethics committee tacked on at the end.
Right.
It's a dedicated social science program led by Professor Joy Zhang, running in parallel with the lab work, specifically looking at the ethical, legal, and social implications, the ELSI issues.
So, social scientists embedded with the biologists.
Essentially, yes. It's about interdisciplinary collaboration from the start, working with academics, civil society groups, industry, policymakers globally.
Yeah.
The idea is to kind of co-create responsible science, not just review it afterwards.
That does sound different. Proactive. What about safety inside the labs? Concrete measures?
They're stressing rigorous safety. For now, all the work is strictly contained in labs. They state very clearly no intention of creating synthetic life.
Right.
And they're building in technical safeguards, things like inducible genetic kill switches.
A kill switch. Explain that. What does that mean practically?
Think of it like a built-in self-destruct mechanism for the synthetic cells. If something goes wrong, or if they needed to ensure containment, they could trigger this switch remotely to eliminate the cells, make them unable to survive outside the lab.
Okay, a biological off switch.
Basically. Plus, they're operating within all the existing regulations while also working proactively to help develop new guidelines as the science evolves.
It's still pretty bold for the Wellcome Trust to fund this, given the potential controversies. How did Dr. Tom Collins there justify it?
His rationale was framed around assessing the cost of inaction.
The cost of not doing it.
Yeah. The thinking is this technology is likely going to be developed somewhere, by someone eventually. By funding it now, openly, within this integrated ethical framework, they believe they can guide its development in as responsible a way as possible.
So better to shape it proactively than let it happen uncontrolled elsewhere.
That seems to be the logic. It's a strategy of proactive engagement.
But even with all these measures, there must still be challenges ahead, right?
Oh, for sure. Huge challenges remain. Getting regulatory frameworks to keep pace with the science is always tough. Public understanding and acceptance is crucial for something this transformative. And just the sheer technical difficulty of working with the human genome shouldn't be underestimated.
Okay, so wrapping up this deep dive.
Right.
It's clear the Synthetic Human Genome Project is, well, a genuine watershed moment.
Absolutely.
It offers these incredible new ways to understand and potentially manipulate life. Transformative possibilities for health, disease, maybe even global challenges like climate change. But, as we've discussed, those benefits come hand in hand with really significant risks and profound responsibilities.
And what stands out, I think, is that integrated approach to ethics they're trying. Weaving the social science right into the technical development, it could be a really important model for how we handle other powerful emerging technologies responsibly.
A model for responsible innovation.
Yeah, that proactive engagement, aiming for real societal benefit while trying to anticipate the pitfalls.
And that quote from Michael Dunn really resonates. Through creating the necessary tools and methods to synthesize a human genome, we will answer questions about our health and disease that we cannot even anticipate yet.
Transforming our understanding of life and well-being. It's powerful because it points to the unknown unknowns, the discoveries we can't even imagine yet.
It really is. It leaves us, and you, the listener, with a pretty profound thought to chew on. We're standing on the edge of an era where creating life, or at least its blueprint, becomes possible through human design.
Yeah.
So what are your thoughts on humanity's role in potentially redesigning the very mechanisms of existence? And maybe more importantly, how do we collectively ensure this incredible power serves the common good and we navigate away from those potential perils?